Well, hello, everybody. My name is Leslie Holland. I'm the UW Fruit Crops Pathologist. And today we are going to be, this is a kickoff webinar of our Advanced Apple Production and Management webinar series. And today we will be discussing rapid or sudden apple decline with Dr. Tami Collum from the USDA ARS. So before we jump into that, I would just like to remind everybody to keep muted during the talk. At the end, you can unmute yourself and ask questions, or also put your questions in the chat box for Q&A at the end of the discussion. We're also going to ask that you hang around till the end to complete a couple survey questions when we wrap up. And I also want to remind you of our remaining webinars for the series. So March 3rd, we have mating disruption, March 24th, bloom thinning, and April 14th, ground management. And registration information can be found on our Wisconsin Fruit News website for each of these webinars. And we do send out frequent reminders to the Apple grower listservs. Additionally, I'm going to put links. Oh, they're already in the chat, it looks like. Thank you, Christelle. And I've got a couple to add as well. And so with that, I think I'm going to go ahead and get us started and turn it on over to our speaker. And while Tami, you are loading your slides, I will go ahead and introduce you. Perfect. Tami Collum is a research plant pathologist with the USDA Agricultural Research Service located at the Appalachian Fruit Research Station in Kearneysville, West Virginia. She began her position in 2021 and is interested in developing sustainable monitoring and management practices for diseases of stone and pome fruit during production and after harvest. Tami received her PhD in cell biology and molecular genetics from the University of Maryland, where her research focuses on virus-plant interactions. After that, she joined the USDA as a postdoctoral researcher at the Appalachian Fruit Research Station, and then at the Foreign Disease Weed Science Research Unit in Frederick, Maryland, where her research focused on diseases of fruit trees, including plum pox and HLB. In her current position, a major focus of this work is investigating rapid apple decline, which will be speaking with us today, and developing scale neutral management practices, which she will discuss in the webinar as well. So Tami, thank you for joining us and take it away.
Great. Thank you very much, Leslie, for that introduction. And I'm happy to be here with you all today to talk about my work with rapid or sudden apple decline. So first, a quick outline for my talk today. I'm going to start a little bit with about an introduction, a little bit about me and my background and what our mission is at the Appalachian Fruit Research Station. Then I'll talk about what is rapid apple decline, some factors that may contribute to rapid apple decline, as well as some ongoing research that I'm starting up at the Appalachian Fruit Research Station, and then some take-home messages about what can be done to prevent rapid apple decline. So as Leslie mentioned, I got my bachelor's degree in microbiology from Kansas State University and a PhD in cell biology and molecular genetics at the University of Maryland, where my background's really in plant virology and pathology. And in my previous research, I've done a lot of work with virus transmission, systemic movement and disease development, and plant responses to disease, particularly in the phloem vascular tissue. And this has been with looking at phloem tissues, has been in fruit trees with looking at plum pox disease and prunus, as well as HLB and citrus. And I just began my current position at the USDA ARS in July of 2021. And in this position, I'll be focusing on diseases of stone and pome fruit trees. So this is the Appalachian Fruit Research Station. So we're located in the panhandle of West Virginia, in Kearneysville, West Virginia. So we're very close, less than an hour away from Northern Virginia. We're close to Maryland and only about a little less than 2 hours from Pennsylvania. So we have a lot of collaborations in that area in the Mid-Atlantic. So our research mission is to conduct fundamental, applied, and developmental research on critical problems of temperate fruit production. And so we have two major focus areas. First, the genetic improvement of fruit crops using advanced genomics and breeding technology, as well as integrated production and automation for temperate fruit crops, which is the group I'm a part of. And we have a lot of scientists with diverse skill sets, including molecular biologists, geneticists, breeders, soil scientists, horticulturists, engineers, entomologists, and I am the sole plant pathologist currently. So in my program at AFRS, my overall research objective is to develop scale-neutral and sustainable monitoring and management practices for diseases of fruit crops. A major focus is rapid apple decline, which I'll spend the rest of this talk talking about today. But I also have projects related to looking for alternatives to antibiotics or fungicides for disease management, particularly fire blight and post-harvest fruit rots. And then I'm also working with our apple and pear geneticists and breeders to look at sources of genetic disease resistance, particularly looking at the USDA's apple and pear germplasm collections and looking for resistance in wild relatives. So when we're thinking about diseases in apple trees and how apple trees respond to stress, I first want to remind everyone that when you're thinking about growing an apple tree, you have to think about both the rootstock as well as the cultivar or the scion. So the genetics and the interactions between these two parts of the tree can influence how an apple tree responds to both biotic stresses as well as abiotic stresses, and they're joined at this graft union. And so the rootstock is important for controlling the size of the tree. And so historically, apple trees were grown on seedling rootstocks, but now we have all these semi-dwarf and dwarf rootstocks that can decrease the size of the tree, which can lead to being able to plant more trees per acre and able to more easily manage the trees and harvest and increase yields. And so when you think about commercial apple production today, you think a lot about these semi-dwarf systems or heading towards these higher density production systems. And so in the semi-dwarf system, these rootstocks, the trees are about 8 to 16 feet tall. The trees are spaced further apart, about 10 feet and sometimes 18 feet between rows. The trees are self-supporting, they don't require any trellises, and you can get about 150 to 242 trees per acre in crop in about 5 to 6 years. You're seeing a lot more high density plantings with these dwarfing rootstocks. The height of the trees is smaller at 6 to 14 feet. They're spaced much closer together. And these trees require support, but you're able to get more trees per acre. Although they do require more manipulation, they require training and pruning and more upfront costs to establish these trellis systems. However, you can get crop earlier in some cases in about 3 years, and you can get higher yields. And why I mention these different apple production systems is rapid apple decline has really been associated with these high density systems in these dwarfing rootstocks. So what is rapid apple decline? So it's also been referred to as sudden apple decline. I tend to call it rapid apple decline because that term was coined by the Pennsylvania Department of Agriculture, which I have a lot of collaborators and work with growers in Pennsylvania. So that's the term I tend to use, but you'll also hear it referred to as sudden apple decline. They both describe the same syndrome that's affecting these dwarfing trees. So this is an emerging problem for apple production in both the US and Canada. And it really started in 2013 with reports in Pennsylvania about an unusual rapid or sudden decline of young dwarf apple trees, particularly in these high density orchards. And so blocks can have a mix of dead, declining, and healthy trees, and they're distributed throughout the block with no obvious pattern. And so here's a picture of one of these trees that's declining. So what are some common trends we're seeing in these trees? They tend to be young. 2 to 8 year old dwarf trees are the most susceptible. We see the most problems on M9 rootstocks, although there are been issues that have been reported on other rootstocks, including B9, G41, and G935. There's several different cultivars that have been reported to have problems. And interestingly, the roots appear to be healthy. We often see these suckers coming out from the trees. Even as they're declining, you see all these healthy root suckers coming out. The leaves of the trees begin to look pale yellow, then reddish, and within 2 weeks, the tree can be dead. And then trees can collapse with a full crop in July to September. The graft union of the tree is affected. You see severe shedding of the bark, and kinkers can be present. And you see necrosis that starts at the graft union and then proceeds up the trunk of the tree. The affected wood is usually solid and not spongy. So RAD has been a particularly costly problem in Pennsylvania in particular. So you're losing trees early and when they're in their prime, or they either are just about to produce their first full crop, or you're losing them before they produce their first full crop. And since they're in these high density production systems, there's all those upfront costs to get the trees in with the trellises, and to have to replace all those declining trees is a significant cost for growers. And I have two examples here from two growers I've talked to in Pennsylvania. One grower had to replace 100% of their trees in 10.75 acres. So that was 8,756 trees. And then they replaced another 5.92 acres of apple blocks. So they calculate their final losses were over $200,000, and that's not even factoring the loss of revenue that would have been produced from this acreage. Another grower in Pennsylvania, they planted 70,000 trees in 2017, and by 2 years later, in 2019, 75% of the trees had collapsed. So as you see, this has been a very serious and costly problem for our growers in Pennsylvania. And we've heard reports from other parts of the country too of growers losing trees to this mysterious syndrome. So particularly in Pennsylvania, where this was first described, this is data from my collaborator, Carrie Peter at Penn State University. And she was really one to first bring attention to this problem. So this is a schematic of her research orchard at the Penn State University Fruit Research and Extension Center. And so here on the left, I'm showing a diagram of her block with declining or dead trees in red. And in 2014, she found that 102 trees, about 9.4% of the block, were declining. This was a block that was planted in 2011 and 2012. And then 3 years later, in 2017, 20% of the block was now dead from the syndrome. And she started seeing symptoms, as early as 2013, 2 years after the block was planted. We have also seen some trees declining at my location at the Appalachian Fruit Research Station. And here's pictures of 2 trees. You can see they have a pretty full crop load, but they're completely dead and declining, some yellowing of the leaves. And so this, as I mentioned, I just started this position in the summer in July of 2021. So I was able to get out there a little bit this season and try to look and characterize some of these trees we're seeing at our location. But I'm hoping to further characterize these and see how many trees we're actually losing in our research blocks at the Appalachian Fruit Research Station this upcoming season. So what contributes to RAD? So the first thing is the cause of RAD is still unknown, but there are several factors that are being investigated and are likely to play a role. And the big take home is that it's likely to be a combination of multiple stressors that are ultimately leading to the tree to collapse and decline. So the factors that are being investigated, there's this question of known and newly discovered apple viruses. There's some look into other potential biotic pathogens, either bacteria or fungus, that could also be contributing. There's a lot of talk about weather-related stresses, particularly severe cold and winter injury that could be causing the collapse of these trees, as well as potential drought events that could be contributing. Researchers are also looking at long-term sublethal effects of certain herbicides that could also contribute to tree stress, as well as potential insect damage from Ambrosia beetles or other insects. There are some biotic factors that have mostly been ruled out. We have not been able to isolate Phytophthora from samples collected to date. In addition, the affected sites have had excellent drainage. There's no evidence that this is due to replant disease. We see decline in both virgin and old orchard land, and fire blight has mostly been ruled out, although I left it with an asterisk because we have not been able to detect their Owenia amylivora bacteria in any of the affected trees. However, there is some ongoing discussion and research into potential latent infections, that if the bacteria is able to get into the rootstocks, if that might be playing some role. So first I'm going to talk about the viruses and how they might be contributing. So the major virus that tends to cause problems in apple trees, particularly in our region, is a tomato ring spot virus. And so this can cause necrosis at the graft union. So this was a first, thing to try to look for to see if there was any tobacco ring spot virus. It usually causes abnormally thick, spongy, orange-colored bark and distinct necrotic line at the scion rootstock union. And the virus is spread by these dagger nematodes, but it can also be transmitted by grafting. And weed management is particularly important for preventing the spread of this virus because it can also infect weeds like dandelions, and those can serve as sources of the virus. But this virus has largely been ruled out, at least in Pennsylvania. It was not found in any of the trees in the rat-affected orchard at Penn State University, was not found in any of the weeds. As far as the other apple viruses go, there are several latent apple viruses, and they're called latent viruses because they don't usually cause any symptoms in commercial apple cultivars. And so these would include apple stem grooving virus, apple stem pitting virus, and apple chlorotic leaf spot virus. And so these are common to all fruit growing regions in Eastern North America. And as I mentioned, they don't normally cause any symptoms. Here, I'm just even showing this is what we call an indicator plant that is very susceptible to the virus. And even then you don't see that strong disease symptoms. They're transmitted by grafting. There's no insect vector. And infected trees can grow adjacent to healthy trees for years. And these viruses can occur in single or mixed infections. And as I mentioned, they haven't really been a problem in commercial apple production. Apple mosaic virus is another virus that can infect apples. Sometimes this is included with the latent apple viruses. Some cultivars are more susceptible, while others show no symptoms at all. And sometimes it can take a couple of years to observe symptoms, and they can be unevenly distributed in the tree canopy. When there are symptoms, they tend to be these yellow chlorosis on the leaves, and it can affect the tree growth and lead to slower growth and lower fruit yields. This virus is also spread by grafting, and there's no insect vector. So there's a question of whether these viruses in combination with other stresses might be playing some role in rapid apple decline. And I want to point out that they're fairly common in apple orchards. This data comes from a survey that was done by researchers at Cornell in 2016 and 2017, looking at orchards in Western New York. The Hudson Valley and the Champlain Valley were selected for virus surveys. And so they tested 24 different orchards. And I want to point out that of all the orchards they tested, they had at least one sample test positive for apple viruses. And you can see here the percentage of samples that tested positive for each of the individual viruses. Apple stem pitting virus and apple chlorotic leaf spot virus were the most common. And as I mentioned, that they can also occur in these mixed infections, and mixed infections of apple stem pitting virus and apple chlorotic leaf spot virus were pretty common, 8% of the samples. Importantly, there were as many trees where none of these viruses were detected, and that's very important for establishing mother trees and for using those virus-free trees to have propagation materials. There's some question of what effect these apple viruses have on the tree. There's some evidence that there's a decrease in yield. These data comes from older papers published in the 70s and 80s, and the amount of yield reduction is pretty variable for the different viruses and different apple cultivars. And a lot of this we don't know for other apple cultivars. If there really is that much reduction in yield, it's potential that they could influence yield. But again, it hasn't been much of a problem in commercial apple production. So there hasn't been a lot of research on this topic. There have been some newly discovered apple viruses, and these have all come from new high throughput sequencing, particularly in these declining trees. So when we first, found all these declining trees, there was people looking for a potential pathogen or cause. And so there was done a lot of high throughput sequencing to try to identify any novel pathogen that might be occurring in these trees. And some new viruses were found. They include citrus concave gum associated virus and apple hammerhead viroid. And they also found several of these latent viruses I mentioned. And so this data comes from trees from Washington State. However, you can see here, they also found these same viruses in trees that appeared perfectly healthy that were asymptomatic. And so there was no clear smoking gun or clear virus that was associated with rapid apple decline. We just know that we're finding a lot of these viruses in the trees. but we also find them in trees that appear perfectly healthy. So there's more work that's needed to understand if these viruses are contributing to RAD. There's also an apple luteovirus that was discovered in Pennsylvania. Similarly to the study in Washington State, this was found using high throughput sequencing, using samples from declining trees in Pennsylvania. And that block I showed schematics of from the Penn State University Fruit Research Extension Center. And a similar story. There was, interestingly, apple luteovirus 1 was found in all the symptomatic declining trees, as well as other apple latent viruses. But it was also found in trees that appeared perfectly healthy, and a high percentage of the healthy looking trees also had all these viruses in them. So it's unclear, again, it's not likely that this virus is causing rapid apple decline, but the fact that these trees have a lot of these apple virus infections in them probably isn't helping with their overall stress. And it could be that a combination of multiple stressors are what's tipping, the scales over the edge so that the tree starts to decline. And interestingly, this is the first report of a luteovirus in apple, and that's interesting to me because it's a phloem-limited virus, and especially looking at these, necrotic issues that we see in apple decline at the graft union. A lot of times, pathogens and infecting the phloem can impact things that are happening at the graft union. So there's more ongoing surveys right now trying to determine how many, how prevalent these apple viruses are throughout the United States in different apple production areas. And this is work that's being done by Oscar Hurtado Gonzalez, who's with USDA ARS in the POM quarantine program. And so he's been collecting tissues from different orchards sites throughout the United States, and we provided him some samples from our location, the Appalachian Fruit Research Station, and he was able to share the results with us. And we do find that at our location, we do see a number of these latent apple viruses, as well as some of the newer ones, particularly this apple hammerhead dwarf virus in our research blocks. And the ones I've highlighted in orange here is the block I showed pictures of before, where I've been looking at and seeing the declining trees at our research station. And you'll note there's some viruses found in those blocks as well. And so, as I mentioned, I've been trying to follow these trees in this block where we're starting to see some decline. So this last season in 2021, I flagged several trees and took samples. We're looking at samples from leaves, petioles, roots, bark, and we're currently isolating RNA and DNA from them. And we're planning to use PCR to test for these apple viruses and other pathogens that may be associated with RAD. And we might also do some sequencing to try to determine what's going on in these trees. In 2022, I'm planning to continue to do more experiments on our block at the Appalachian Fruit Research Station to follow the natural incidence of declining trees and take samples at 2 different time points in April to early June, and then again later in the season in August and September. And part of the reason I'm using multiple sampling time points is that it's known that virus titers can vary a lot in fruit trees. And so the virus titer can vary from tissue to tissue, as well as during different seasons, seasonal time points. And this can be a challenge for detection. And a lot of this we actually don't have a lot of data on. This is data from one virus, tobacco ring spot virus, was recently published by Scott Harper. And you can see that there's different fluctuations in the amount of virus you can detect across the different time points in the season. But we don't have a lot of this kind of information, especially for the newer apple viruses. So that's one of the areas I'm trying to look into, is try to be able to further characterize, when these viruses are most accumulating in the trees and how that changes over different seasons. And that's very important for being able to detect these new viruses and make sure that we're able to provide tested virus materials for propagation. So what can be done to limit these viruses? So unfortunately, once a tree is infected by a virus, there's really no direct way to cure the tree or to combat the virus besides removing the tree. So you want to try to prevent them from getting infected in the first place. You can test the soil for nematodes prior to planting, and a good weed management program, particularly looking at weeds for control of tobacco ring spot virus, can be important for preventing its spread. And then the really important thing is to try to get propagation materials that have been tested for viruses, and make sure if you have a tree that you're using for budwood, that you test it to try to make sure you're not continue to propagate these viruses by grafting. So another key factor that has been talked about a lot in regards to rapid apple decline is this potential role of winter injury. There's several folks have proposed that really that this just comes down to winter injury, and that there might not be other factors at play. We've just been seeing a lot more severe weather events that is leading to the decline of these trees. And so winter injury most often occurs in extreme low temperatures, especially when low temperatures follow warm temperatures. And the capacity of apple trees to survive low temperatures is influenced again by the scion as well as the cultivar. And so the scion or the cultivar as well as the rootstock. So when trees are damaged by winter injury, you can get shoot death and dieback, and it happens more often in young trees. So you often see this symptom development in trees that are damaged by winter injury. You see this browning of the phloem and cambium under the bark in irregular patches or streaks. You can also see the bark may split and peel. And you can also get cankers forming over the xylem, which can be infected by wood decay fungi. After winter injury occurs, you can continue to see new symptoms and dieback even for a couple of years after the particular severe weather event. So we were interested in looking at the trees at the Appalachian Fruit Research Station that I had pointed out. I saw that were declining in our blocks. And so I started looking at cross sections of branches to see if we saw any browning of the phloem or the cambium in these trees. And indeed we do. So this is some representative pictures from branch cross sections of declining trees. We had 3 different declining trees as well as 1 healthy control that we cut up. You do see some browning in the healthy control, but we saw more in the declining trees. And so I was able to quantify that. And so how I did that is I basically converted these images to a binary black and white image, and then use a software program to quantify the amount of dark pigmentation. And so I did find significantly more this dark browning in the declining trees as compared to the control. But it could be that winter injury is playing a role at the trees that I saw declining at our location. However, it's hard to completely determine because pathogens and other stresses can also cause browning of phloem tissues. So the question is winter injury the cause of RAD? So we do see browning of the phloem and cambium in RAD-affected orchards. And so this is another picture from a cross-section from a RAD-affected orchard. You can see a lot of browning in this tree. However, we're not completely convinced that winter injury is the only cause of rapid apple decline, because the pattern of declining trees is not always consistent with winter injury alone, particularly in Pennsylvania. So the tree loss was not always limited to the coldest parts of the orchard, and particularly in the research block at Penn State University, we saw a decline of trees, Fuji, Gala, and Golden Delicious were all affected, but Crimson Crisp was not, despite being evenly distributed throughout the 3 acres. If this decline was only due to winter injury, we'd expect to have seen more loss in Crimson Crisp as well, since they were all on M9 rootstocks. So it's likely that winter injury is very likely to play a role and may be responsible for declining trees in certain orchards. But there's also likely to be additional factors that are causing stress and weakening the trees. And it might not always be the same factors at different orchard sites. And so one question I've been interested in trying to address is if it could be winter injury as well as these viruses. And so I'm interested in trying to look more at this question of, do virus infections make apple trees more susceptible to winter injury? And there's very limited data on this. I was able to dig up one older paper from 1998 where they were able to show that trees, so this first bar is the control of trees that weren't infected with virus. The next two bars are trees that were infected by virus, and the darker colors are trees that were injured with the lines or killed in black. And you can see for Macintosh trees, apple trees, the controls were not injured or killed as often as trees that were infected by viruses. And Macintosh is known to be a cultivar that's more resistant to frost damage. But for the other cultivars that this experiment tested, the control trees and the virus infected trees, basically the majority of them were damaged by either injured or killed from frost damage. So there wasn't a clear difference, but it does suggest that there might be some impact of virus infection, particularly in trees that are already pretty cold hardy. And I'm interested in trying to understand more about this, particularly for some of these newly discovered apple viruses where we really don't know much about their biology or how they're impacting the tree. So then there's some question of if it's not viruses, it could also be some other factor or stress in combination with winter injury. And there's been several hypotheses put out, including drought stress or herbicide injury, as well as just the stress of this high density production system. So we think, we're growing trees closer together and they have to compete for water and nutrients and have these smaller root systems. And, we're pushing them hard to produce fruit early. So maybe it's just the stress of the system along with winter injury events that are leading to decline. So how would you go about limiting winter injury? So the choice of the variety of rootstock can influence how susceptible the tree is to damage during the winter. You can avoid poorly drained and low-lying areas, avoid fertilizing late in the season, and avoid pruning late when you, that's important to allow the tree to harden off so that doesn't continue growing late into the season. And you can keep crop loads lower to try to minimize tree stress. You also can paint the trunks white or use guards to prevent southwest injury. Another potential stress I mentioned was herbicide injury. So there are also some strategies that can be used to try to prevent any effects from herbicide application. And so this has been seen that this herbicide application has potential wounding particularly young trees and increasing their susceptibility to secondary infection and winter injury. So you can use trunk shields to protect trees less than 3 years old and use drift inhibitor in the spray mixture to minimize the number of small droplets. Use hooded boom sprayer to minimize the bounce back from bare soil, and never apply with controlled droplet applications that generate superfine droplets. And you want to avoid using glyphosate for sucker control because that can then allow it to be taken up into the tree and reduce winter hardiness. Another factor that has been investigated and of being potentially associated with RAD is these Ambrosia beetles. And so we found that there was presence of borer holes in some declining trees. It's been observed that trees in poor health had a greater number of beetle holes, but the borer issues were not identified in all orchards with tree collapse. It's known that Ambrosia beetles are attracted to ethanol produced by stressed trees. So it's likely that the beetles came in after the tree was already stressed. Although interestingly, it has been observed that some trees that looked healthy also have beetles. So it's possible the beetles may be able to detect the stressed trees before we can. And that's an interesting direction for research to try to develop earlier detection of these declining trees. There's also been the suggestion of other pathogens that might be involved. There's researchers looking at fungal species that have been associated or isolated from RAD trees. However, this hasn't been really consistent. There's some look at fungal canker and wood rot pathogens. However, these usually cause a slow decline rather than a sudden decline. And it's likely to be that a lot of these fungal species that we're finding in rad trees are secondary or opportunistic infections. However, they could be contributing to overall tree stress. So areas I'm trying to look at to try to get more information about what's occurring in these trees is I'm really interested in trying to understand how these apple viruses might be playing a role, getting a better understanding of their distribution and seasonal accumulation, particularly for these newly discovered apple viruses, and looking at different tissues and seasonal time points and different scion and rootstock combinations to try to determine when these viruses are most accumulating, and this is important for accurate detection. I'm also wanting to look more at this potential interaction between stress caused from virus infections and the abiotic stress from winter injury, of whether these virus infection makes trees more susceptible to winter injury, particularly in mixed infections or with these newly discovered apple viruses. I'm also interested in looking at host responses during rapid apple decline, because regardless of whether it's a biotic stress or abiotic stresses, or likely a combination of both that are leading the trees to decline, by looking at the host, we might be able to identify some markers that are able to predict which trees are going to decline. And so there's really not that much known about the genes or pathways that are altered in apple trees during this rapid apple decline. So knowledge of these host responses could help with early detection, help with developing early intervention strategies, or the development or identification of resistant cultivars. And so I'd like to use high throughput sequencing to try to characterize these host responses in declining versus healthy apple trees. And then the goal of this research is to evaluate the apple host genes for if we can determine any early molecular markers of RAD that might enable us to identify trees early, and then look at potential management strategies that could help prevent these trees from further declining. So I've identified a number of orchards that I'm planning to look at in this upcoming season, mostly in Pennsylvania. There's 5 commercial orchards there that my collaborator, Carrie Peter, has been following for a number of years, as well as the research orchards, both at Penn State and then at my location at the Appalachian Fruit Research Station. And so we're planning on flagging declining trees and following them throughout the growing season. And then we'll also use healthy trees as a control, and looking at these different time points in the season, an earlier time point in April to early June, and then later in August to September. I'm also working with our horticulturists at the Appalachian Fruit Research Station to put in some new apple tree blocks, and we're looking at a couple different rootstocks. And these rootstocks were chosen because they're part of the NC 140 multi-state research project to evaluate rootstock performance, as rootstocks, the rootstock performance can be variable across different field sites. And so we have a number of dwarfing rootstocks, as well as a couple semi-dwarf rootstocks, and they were chosen for different horticulture properties, as well as certain of these rootstocks have been associated with RAD, particularly M9, as well as some have been, particularly G935 has been reported to be sensitive to latent viruses. So we're interested to see how these trees do at our location, and we're going to be monitoring these trees as they go in and see if we're getting any of these early losses when we're planting these in a high density system. And so the goal is to be able to use this block to both investigate biotic as well as abiotic stresses and scion rootstock interactions that affect apple tree growth. So there's a lot of things that we still don't know about rapid apple decline, and it can be kind of confusing because there's all these factors that are likely playing some role, and we're still doing a lot of research to try to really get at the bottom of what how much each of these different factors are contributing to tree decline and what their interactions are with each other. But I want to leave you with this take-home message that there are some things in our control, and the big take-home message is minimize tree stress. So all these factors that could be contributing to RAD are causing stress on the tree, And it's not likely that one of these is responsible alone, but that multiple stressors are coming together to cause this decline. And so what can you do to minimize tree stress? You can test soil for nematodes and nutrient levels prior to planting. You can use virus tested propagation materials. You want to make sure you have proper planting depth and have the graft union 2 to 3 inches above the ground. We found that if it's higher, you can be a problem and have provide opportunistic places for insects to feed. It can lead to problems. Consider irrigation, especially if you have trees on M9 rootstocks. You want to maintain proper nutrient program throughout the year based on soil and leaf analysis, as well as limit winter injury. So you can avoid fertilizing or pruning late in the season, paint the trunks white or use white tree guards to prevent southwest injury, use trunk shields to protect trees less than 3 years old from herbicide sprays, and protect the graft unions, particularly from rodent damage, as well as look at season-long disease and insect management. And so I want to thank all the people who helped contribute to this work, particularly Carrie Peter, who's a collaborator at Penn State University, who has really described this syndrome as occurring and identified it in Pennsylvania, as well as Lisa Tang, who's helping me with the orchards we have at the Appalachian Fruit Research Station, my technician, Brianne Evans, and as well as Oscar Hurtado Gonzalez, who's doing some virus surveys and was able to share with me the data from the viruses we have at the Appalachian Fruit Research Station. So with that, I'd be happy to try to answer any questions.
Go right ahead.
Thanks. I have 4 quick questions. The first, have they found this with in blocks that have tree guards, solid tree guards in place? Secondly, when you were doing the cross section looking for browning for winter injury, what age were those branches that you were checking? Were they 1 or 2 year old or whatever wood? Third, when Dan Donahue last week, I think that's his name, up at Cornell, talked at the last week's Cornell IPM thing about virus load, and I'm not sure if he was referring to virus, the amount of individual viruses, if there's a way to measure the quantity of virus, or if he was just referring to the number of viruses possible. It's associating the higher virus load with more symptomology. And lastly, you talk about having guards on 3-year-old and younger trees. There's this argument that high density trees never have what we would call mature bark, that a 9 or 10 year old bud 9 or whatever could still have immature looking and immature behaving bark. So what do you have responses to those questions?
Okay, I'll try to address each of those, and then you might have to help me with some of them, make sure I answer them all. So you're talking about the branches that I got from those trees. This was kind of a preliminary look, and we actually were taking the whole tree down. So we took off all the branches of those trees. Those were older trees. I think they're more 9 or 10 years on bud 9. So we just basically cut every branch down and looked at them all. I probably in the future will try to be more, we might be able to separate out some of those, but that was kind of a thought of, I was working with our horticulturists to just kind of take a quick look to see if we were seeing any phloem damage or browning in those trees. I think the next question was about, or one of the questions is about the amount of virus load.
Right.
So there are ways to test the amount of virus in the tree. I'm not familiar with the particular presentation you mentioned, but you can distinguish different viruses from each other using PCR tests, and using quantitative PCR, you can determine the amount of viruses that are accumulating in the tree. But there's also other tests for viruses that like ELISA-based, where you're just kind of getting a yes/no answer. Does the tree have virus or doesn't it? And then a lot of the virus characterization, particularly in like the home quarantine program or Clean Plant Network, they use sequencing to try to look at all the viruses that they're finding in a tree. So it kind of depends on, you know, how they were doing the study to determine, you know, virus load. I'm not sure exactly what was being done in that work.
Sure. Do you think
Yes.
John, could you hold on a second? Can we take other questions? Want to make sure that everybody has a chance to take questions. Sure. Thank you.
Thanks, Christelle. Yep, there's another question in the chat from Scott. Are there any official or unofficial projects being considered to survey grower orchards for RADSAT in the upper Midwest states?
I am not aware of any. I am definitely interested in collaborations. Once I get my AFRS permits in place that I can receive samples that might be infected, I would be very open to receiving samples from the upper Midwest. If there's anybody interested in collaborating on that, I'm starting in the region I am, just because, especially with our current travel restrictions, it's easier for me to get to those areas and start with those trees. But I would definitely be interested in expanding. I think that the virus surveys that Oscar is conducting, I believe those are a national survey. I know, you know, people from our region have provided samples. I'm not sure if there's been, if anyone's been in contact with him to provide samples, but I'm sure he would, you know, be open to that too. And that's data that's unpublished yet, but I know he started collecting samples from us in 2020 as trying to get a good idea of, you know, how prevalent these latent viruses and these newly discovered viruses are in apple production regions throughout the United States.
Yep. Yeah. Thank you, Tami. And when Tami and I first met, and part of the reason I invited her to speak today was the talk of this RADSAT for Apple. So I think, yeah, collaborations is open and welcome. So hopefully we'll get you to Wisconsin at some point and we can start looking. On that note, Gary asked, has any apple trees been detected in Minnesota or Wisconsin? So I don't know if you know, I know from nothing official, right? Maybe seeing some symptoms on my end, but I've not heard anything official, but I'd love to know what you've heard.
Yeah, so I've heard, I haven't heard anything from those regions besides, you know, just talking to you, that you had mentioned that, you know, people were interested. I guess the areas we've mostly been seeing, it was described in Pennsylvania in 2013. There's been reports in New York. I know there's researchers at North Carolina State that have been involved, and some reports in North Carolina. There's been reports from Ontario, Canada. As well as I think there's some researchers in the University of Massachusetts that have been involved. And then on the West Coast, I know that they are seeing a little bit different symptoms and trends. A lot of those tend to be on the rootstock that's sensitive to latent viruses. So it's not quite clear if it's the exact same stressors that are going into the problems they're seeing. But yeah, those are the regions I've heard, mostly Pennsylvania, we've seen in West Virginia, North Carolina, some in New York is mostly where I've been hearing about it.
Excellent.
I just put a comment, but if you have observed something that looks like rad or sad in your orchard, could you please say here, we're not gonna identify anybody. You don't have to, like, it doesn't mean that you had it in your orchard if you're uncomfortable with that. But if you've seen it anywhere in Minnesota or in Wisconsin or in your location, could you please just say yes if you've seen it in the chat?
Yes.
John, you said yes?
Yes, unfortunately, I did.
Okay.
And I invite you, if you would not like to express that here, of course, you can reach out to me over email. I've got Tami's contact information. And again, Tami and I have spoken in the past about collaborating, or at least, you know, extending samples to support the research on this disease. Oh, Steve also said yes.
I guess, John, go ahead if you have any more questions. Oh, no, there's another one. Sorry. Go ahead, Leslie.
Oh, I've seen something we thought may have been rad in Maritime Canada, but ultimately I think it was something else. So, and I think, yeah, that's the difficult part with this, right, is trying to diagnose, because a lot of these symptoms have a lot of overlap with other, you know, abiotic or biotic things that we see. So it's always good to try and contact one of us. Oh, could you answer John's first question regarding tree guards?
So I know that my collaborator, Carrie Peter, is doing a trial with tree guards in particular, trying to determine, how much they can prevent seeing these rad symptoms. I don't think she has results from that yet. As far as whether it was observed in orchards with tree guards, I think that there were some growers that were using tree guards, but I'm still getting caught up to speed since I just started, getting out there this last season. So I can try to find out the answer for you and get back to Leslie and have her pass along that information if that's, yeah.
That's great. Thank you, Tami. Other questions?
Go ahead, John.
Oh, that's right. We had some. Yep.
I was just going to share the situation that we saw that we thought might be RAD, which occurred in 2019 on Evercrisp on G30s. And these trees were planted in 2017. And in 2019, they had a big crop. And between July and August, they lost 10%, which was only 40 trees. But 10% collapse, just in a classic like you described. And when we looked at the bark on those, I graded the bark on the remaining 380 trees in October of that year, and basically told him he was probably going to lose most of the rest. And we sort of, there wasn't a clear herbicide damage, but those are the only G30s he has in that orchard. And so at any rate, in the next year, in 2020, I asked him to not have any fruit on those trees, to defruit them. And those were the most gorgeous trees in the entire orchard. This past summer, then they had a light crop. So they have not suffered any more death. I think one or two more trees have died in the ensuing 2 years. And so me telling him that he'd likely lose most of those within the next 4 or 5 years so far has not come true. Thanks.
Thanks, John. There's another question in the chat. Does it always affect the whole tree or maybe just a branch?
So it tends to affect the whole tree because it's really a problem at the graft union that spreads up. So once there's the problem with that graft union, the whole tree collapse. I haven't seen any cases where, just a branch is affected. Yeah.
It's a good distinction. And thank you, Sarah, too, for your comment in the chat about symptoms you've been seeing on M111 and B9, and the M7s are fine. Something I thought about, Tami, and again, continue rolling in with the questions in the chat and unmuting yourselves, but in an early slide, you showed the research plot at Carrie Peter, the apple trees, and it looking maybe 3 years later at the increased incidence, it seemed like a lot of those trees were really sectioned to one side of the orchard. So I wonder if that there are any features or characteristics of that side of the orchard that might have played a role in why we saw that incidence jump up there.
Yeah, so far we haven't been able to determine an underlying trend of a certain part of things that are in certain part of orchard. Just talking to growers in Pennsylvania, the different orchards, there's some that have said it's mostly the trees in the lower lying areas that are affected, where others in their orchard, it's the higher elevation trees that are affected. It seems to be pretty variable. And it doesn't always, there's some, you can see from the plot I showed of Carrie's that there's some that are trees, next to each other seem to go, but it's also, they'll pop up, kind of randomly in the orchard. So there's still kind of a question of, is it really spreading at all from tree to tree? Because if it is these, viruses, there is the potential for root grafts if they're contributing, but in general, there'll be trees that are right next to each other and one's fine and, the other one isn't. So it doesn't necessarily seem to be that's a clear pattern either. So we really haven't determined a clear, pattern. I would say that things that affect other stresses like winter injury, or if you have drainage issues in certain areas, I think just the more tree stress you have in certain areas of your orchard, you're probably going to see more issues with rad there too, because it's probably a combination of, multiple stressors coming together to really cause the tree to decline.
Yeah, I completely agree. Yeah, predisposition, we have to talk about it more with these perennial fruit crops. So a couple more questions in the chat. Rad and organic versus conventional orchards. It's a great question.
I don't know if there's been any studies to really characterize this. I know the ones in Pennsylvania I've been looking at are mostly commercial orchards. I can't think off the top of my head. I think that, I've heard, I guess the second question too is if it's like hobby style orchards. I have heard from people in West Virginia and Pennsylvania that they say that they're seeing these issues in their home orchards as well, smaller numbers of trees. But I'm not sure if there's really been a study to look at differences between organic versus conventional orchards and the incidence of this.
Okay, that's great to know. I'm going to launch a poll as we get kind of close to the hour, if folks wouldn't mind completing that, but also still entering or unmuting yourself for any last questions for our speaker, Tami.
There is one more question, Tami. Is there any evidence yet that this affects smaller home style or hobby style orchards, or is it a concern only for high density orchards? You mentioned that at the beginning, but.
Yeah, so it's been prominently found in the dwarfing rootstocks in high density orchards. I've heard, anecdotally talking to people that they think they're seeing this in their home orchard, but I haven't been out there to look at it. And there's a number of things that can look similar, particularly in our region. We do have a lot of dagger nematodes, and tobacco ring spot virus can cause issues at graft union. So it's important to rule out things like that first before determining that it's, necessarily this new syndrome rad. But, so I would say to try to rule out the obvious, like, is it tobacco ring spot virus? Is it something else that could have, and it's kind of rad as the things that can't be explained by the usual culprits, and is likely a combination of these different stressors.
I think apple growers especially are well aware of what's going on when you have multiple stressors because of colony collapse disorder in honeybees. So I think that's, if you were talking to other growers that don't use honeybees, maybe they wouldn't know, but that's exactly when you started with your presentation.
Yeah.
With multiple factors, stressors that are all maybe a little bit contributing, and it's all together. It's exactly the same for colony collapse disorder in honeybees. So
yeah, I think and I've heard Carrie, like my collaborator, Kara Peter, who really first described this, she used that example a lot of it. Is very much like a colony collapse, that we're seeing all these different stressors coming together. And I'm hoping to try to further detangle, how they might be interacting with each other, or, certain biotic factors make trees more, susceptible to other stresses too. But it's still complex, and it's difficult to detangle these multiple factors.
Yep, exactly. There was a question from Lena, but that's something about going back to a slide, and we're at time now, but I put in again in the chat the link to where the recording will be posted. So please feel free to go back to the recording, and you can at your leisure go back to the slides you are interested in looking at, or if you couldn't hear at the beginning, we had issues with people that couldn't really hear well, please go to the recording and you'll be able to get everything you need from there.
Yep, in addition to those websites or links, there's also links where you can register for future webinars, as I mentioned, coming up later over the next couple of months. Thank you again for answering the poll. I'm going to go ahead and end that. And again, let's thank our speaker, Tami Collum from the USDA. We really appreciate your time today. I learned a lot. And so again, thank you. And thank you all for attending. And we hope to see you at the next one.